Good morning, everyone. Thank you for giving me the opportunity to present Fastox Pharma, where I am responsible for business development, marketing, and communication. Let me see how that will work. Okay, so it all started with the observation that although botulinum toxin are the #1 treatment in medical aesthetics, it has also its drawback. It takes a few days for the toxin to kick in, and then after 1 month, where the efficacy is maximum, it's starting slowly to decrease, and the duration is only 3 to 5 months. It's, of course, an issue in aesthetics, where patients can be used to the filler's duration, where you have an immediate effect, and some longevity from 6 to 18 months, but it's also an issue in therapeutics, where patients will re-experience symptoms before they get the next treatments, and this will impact their quality of life. So companies have been looking at ways to increase the duration of toxin, and of course injecting higher dose is a very quick and effective way to do it, but very quickly we reach a plateau effect where you will inject more toxin, but then you won't have any additional efficacy, and you can have additional side effects, and then it's not really cheap for the patient as well. So what we have been doing in Fastox is that we have a technology, the last technology, and our lead candidate, which is boosting the performance of botulinum toxin A. We enable the toxin to enter more efficiently into the neuron. We have been demonstrating this effect in 2 well-known animal models, the DAS, the digit abduction score, and the free running wheel. And we have also demonstrated this boosting effect on the 3 toxins of reference in the market today: Botox, Dysport, and Xeomin. We have also demonstrated that we can boost the effect of the first liquid toxin that was launched in Europe, that is Alluzience, last year. It's, of course, patented, and we are free to operate. So let me show you the result that we get. So here you see a typical activities curve of the DAS, so the digit abduction score in rats, where you see in gray the activity of Botox alone. So as you can see, after the injection, the efficacy starts a few hours, 8 hours in rats, and then it will slowly increase, reach a peak effect, and then decrease, and 8 days after the injection, you are back to grade 1, and a few days after, you are back with no efficacy. When we combine our product, FTP501, with Botox, you get the second curve. And as you can see, we have higher response rates at each time point after the injection. We start quicker, and we are more effective, and we last longer. And we can increase the duration of the toxin by 50%. If we go even further, and we optimize the dose of Botox, which is basically that we inject more units of Botox, and we combine it with our FTP501, we're able to go even further by being even more quick, higher, and we can reach 100% additional duration. So how does that work? Let's imagine that the toxin are people trying to enter into the neuron by a door to do their job, which is cleaving SNAP25. There is only a limited number of doors for the toxin to enter into the neuron. Let's say there is 1 door. And then because of the inhibition effect of the toxin, at some point the door will close, leaving the toxin outside of the neuron. If we inject more toxin, obviously you will have more people trying to enter into that very same door. So you will have a better effect with more toxin inside the neuron, but at some point you will be limited, and then more toxin will be free into the neuromodulating junction. With Fastox, what we enable is that FTP501 will open more doors into the neuron for the toxin to enter. So with the exact same dose of the toxin, you will have more toxin inside the neuron to do its job. We think that a combination of our product, FTP501, with the toxin A will be uniquely positioned into the market because we can act on both onset and duration, both in aesthetics and in therapeutics as well. Where we are today, so we are finalizing the preparation of our first clinical trial in humans It will be a phase 1/2 in glabellar lines in Europe, where we will be able to assess as well as the safety of our combination and our product alone, but also the efficacy on glabellar lines and maximum flow of the combination compared to the toxin alone. We are hoping to enter into this clinical phase at the end of the year. And then, of course, in the meantime, we will, of course, start the development in therapeutics. We believe that because this phase 1/2 can be considered as the phase 2, it could be easy to go, faster to get into the market with a phase 3 afterwards. So who are we? So we are 3 founders. My co-founders are engineers by training. Jean-Marc is specialized in immunology as well afterwards, and Michael in cell biology. We are also supported by an expert in toxin development. Philippe Picot was the former VP for R&D in neuroscience at Hoepffner. We are also very lucky to be supported by key experts in both medical aesthetics, you will recognize the name, of course, and in therapeutics with Professor Garcias, who is a KOL in neurorehabilitation. We are also very lucky to be supported by Gianpietro Schiavo, who is into cell biology and the mechanism of action of toxin. He was the former president of the Toxin International Association. And that's it. Thank you. Do you have questions or do?
We do.
Yeah,
We all have questions for you. Should I start?
Yes, please
Great. Well, thank you for the nice presentation. I'm interested to understand a little more what will be required from a regulatory perspective. When you think about your studies, can you describe a bit more what the expectations will be in order to get a claim that coupled with the toxin, you outperform the toxin alone?
Well, basically, the requirements for the toxin, it will be a toxin combined with FTP501. So the requirements from the FDA will be the same as the other toxin. There is no specific requirement for us to compare a toxin with the activity of another toxin. Mm-hmm. Of course, this is something that you will obviously want to do if you want to claim that you are more effective. So you can design studies, whether into the development plan or afterwards, to compare your combination effect with the toxin alone or with competitors, with, on specific endpoints such as the onset, the response rate, and the duration, of course.
And is that your plan, to go out with an indication or claim that says we're better together?
Well, yes, obviously. Yeah, with a long duration claim, obviously.
Yeah.
Yes, definitely.
Can I pick on that question, the regulatory part? Well, right before I say that, is also fantastic people. I've had the pleasure to work with Natalene for a long, long time. And she's the real thing. And the team there, my predecessor is on your board, and one of the men I admire the most. So congratulations.
Thank you.
But picking up on Karen's question on the regulatory, you showed the activity of Botox in a curve. There was a slide called Fastox boosts significantly
yes.
Botox.
Yes.
And then you showed Botox, and then you showed your product combination. Do you have a slide where you showed just your product? Does your product have an activity of itself?
No. So in rats, of course, we are still in rats right now. We haven't been shown, FTP501 has no activity by itself. So it's kind of an ingredient, but it's an active ingredient. So we think that there would be no need to have a specific focus during our development of FTP501 alone, meaning that there will be no harm in the phase 3 to compare with FTP501 alone.
You don't think the FDA is going to ask you for that?
So this is what we are going to show as well, and we hope that we will demonstrate that we don't have any activity by itself during the phase 1/2, where of course the safety arm, the phase 1 arm, will be also to show the safety and potential efficacy of FTP501 by itself. We are quite confident that there will be no, but we will prove this during the phase 1.
The regulatory piece seems like one of the big questions
of
for
course,
sure.
Yeah.
Yep.
Yeah. Maybe Irene.
Well, and I would say that also on the IP front, it sounds like you have a terrific position on your portion of the molecule, but what do you envision as your challenges for going into the combination world between the different products, and how you would actually be able to claim IP around that, given that the efficacy is actually being delivered by the toxin, not by your product? You have the longevity piece.
Well, IRP is covering the combination of any post-synaptic inhibitor with toxin. So, and FTP501 is a post-synaptic inhibitor. So I think from our point of view, because we've discovered these, let's say, we haven't seen any work on that ever. I mean, the freedom to operate is pretty clear. So we truly believe that there is really no risk of us having some kind of patent infringement or some competition within that post-synaptic inhibitor group. So,
Thank you.
Ask a question that is a little bit outside of regulatory. So every product, we want to say it works faster and longer and less side effects. Okay, so you show there that you're going to work faster and longer. But the side effects that we know that toxins have, and they can be very, very bad, you know, what are you going to do? I mean, do you have an antidote? Because if it works, you know, if the side effect is going to work faster and longer and you
better.
can't avoid it, and well, but the side effect is not your product. It's going to be the toxin. So how do you correct that?
good question. Well, basically, we enable, I mean, first, we enable more toxin to be, to get inside the neuron, meaning that there will be less toxin in the neuromodulator junction that is able to spread somewhere. So possibly we can have less side effects because of this, because there's more toxin internalized into the neuron. And then what we think is that the injectors, they would, I mean, they know how to inject toxin and how to modulate its effect according to the desired effect, basically. I mean, they know how to modulate its efficacy. And so it would be the same. I mean, they would be using the combined product the same way that they are using the toxin. But of course, there is no antidote to FTP501, as there is no antidote for toxin today, basically. So this doesn't change.
But well answered. Yep.
Congratulations. I think that this is
Thank you.
Yeah, thank you very much.